biodecayed clade mycota, okay? And this is alderescent. So this is the big group. It contains almost all of the fungi other than the chytrids. And so they've lost their flagella. You can say either way, okay? That's an apomorphic. We know that they came from an ancestry with flagella, okay? Because that's pretty widespread in the eukaryotes, and also the basal group here, chytrids, they have flagella, and DNA evidence holds all these things together. And furthermore, plasmogamy and karyogamy are separate, okay, in time. So I put this up here as dikaryotic state present, okay, N plus N state is present. The chytrids don't have anything that is dikaryotic, but the rest of them do. Now, these groups will still differ to the extent of the dikaryotic state, and that's going to come up a little bit later. So this here just means that some structure, some cells in their cycle can be dikaryotic, right? And so within here, this is once again another probably paraphyletic group, but we're still going to treat it. I'm not going to give it any sort of apomorphies because it's in the present. Right now, I know some fungi people up at UC Berkeley, and they're starting to divide this up. All right, but we'll still keep it for simplicity. Something called zygomycota, and they're commonly called zygote fungi. So I put it up here. We're still going to have a branch. Okay, zygomycota, all right? But beware that this is probably paraphyletic, all right? And they may be, and well, I say here maybe, but right now it's starting to be pretty certain that they are, because some other groups are being divided off. They are just like the chytrids, non-septate. So they do produce septa in reproductive structures, but that's the only place. The vegetative mycelium, which is below ground most of the time, growing in the substrate, they will have no septa, all right? But that is plesiomorphic for this group, because these chytrids also are non-septate, okay? So these will produce, okay, so the limited, when plasmogamy and karyogamy happen, it's the dikaryotic state is very limited to something called a zygosporangium. And so it's just one structure called a zygosporangium. Unfortunately, your textbook probably calls it zygospores. If it does, I really don't like that word because it's not a spore. It's but that's an old word that they came up with, but it's really a zygosporangium. And this is the only part of the fungus that will ever be dikaryotic. So it's limited dikaryotic to just one particular thing. Right? And a lot of these mostly produce asexually. So it's rare that they undergo sexual reproduction. They do sometimes, but most of the time they produce asexual spores by sending up a stalk like this. That's bread mold. And so there's a stalk. And up here is where the sporangium is. Usually this is what's sticking up above the bread. And they will produce, by mitosis, asexual spores that we're going to call sporangiospores. Okay? So these are produced by mitosis. So there's no mixing of genes at all. So the genetic component of the spores is exactly the same as the parent fungus that they came from. Okay? And of course, the parent fungus is haploid. Okay, haploid. Okay, haploid. Okay, so these are different. This is bread mold, and this here is something else. What is it? Slobium? No, what is the name of that? This stuff here grows on cow poop. Okay, and really interesting. This is sporangium up here. It's sporangium 4. So the fungus is down in the cow poop, and they'll set up these. And this is photosensitive. Okay, what will happen is this will build up pressure on the inside, and it will explosively shoot off all the spores, okay? Because they're in grass. I mean, that's where cows poop, right? Okay? And grass is in the way, and therefore they try to find, they're photosensitive so that they will align themselves where there's light coming through the grass and shoot the spores way out, okay? And that group there. But nonetheless, these are sporangial spores, okay? And this is a typical life cycle. We'll go through a life cycle of bread mold, okay? This is rhizopus. This is what you're going to see in the lab too. So most of the time, they are reproducing asexually, okay? Haploid mycelium sends up a sporangial spore with a bag up here that is called a sporangium. And by mitosis, it will produce spores. The asexual spores are released, they're haploid, and they will grow into a new haploid mycelium. However, under stressful conditions, usually when the food supply is disappearing, you have two different mating types, haploid, okay, plus strain, minus strain. They will grow alongside each other and start sending out a hypha, which is called a conjugating hypha. Conjugating hypha. They're attracted toward each other, and these are gametangium. Okay, so we're using the same word, that they produce gametes. But in this case, the gametes are not open cells; they're nuclei. This is cenocytic. Okay, so first of all, they will come into contact. So we have a gametangium from the minus strand, gametangium from the plus strand. Then they will form a septum, okay, right there. They'll form a septum so that the gametangium is now separated from the conjugating hypha, okay? So that under the microscope, you would first of all see 1, 2, 3, 4 structures, okay? This is conjugating hypha. That is the gametangium from the minus strain. And then the gametangium from the plus strand, and conjugating hypha right there. These are separated, all right? Then this wall between the two gametangia, they'll break down so that we now have plasmogamy, okay? So this has not just one nucleus in it, but many nuclei, and they form then what is called a zygosporangium. So change that. This is called a zygospore, and it's a shame that that's the case, because it is not a spore. It's a sporangium, okay? And this is now dikaryotic. It's only the zygosporangium in this group that is dikaryotic, okay? And this may, how long it lasts as a dikaryotic state is still relatively short, but still pretty spread out compared to what you have in animals and plants. Then these nuclei will undergo karyogamy to become zygotes, okay? And this zygosporangium a lot of times will form a very, very hard, resistant cell wall, okay, around it. And the rest of the fungus can die off, and these zygosporangiums can be around for quite some time until new, better conditions come up. So here we have zygotes. They will undergo, these are diploid, okay? Diploid. They will undergo meiosis to form haploid nuclei, which are now genetically mixed, because we have crossing over, we have independent assortment, and all those good things that happens with meiosis. Out of that zygosporangium, it will send up something that looks just like the asexual sporangium. Okay, no difference in appearance. However, these have been genetically mixed up, right? These nuclei will now migrate up into the sporangium, where they will go through several rounds of mitosis. We still call these myospores, okay? They're still sexual spores, but any one spore that we had, or spore nucleus, we now increase the numbers of that particular spore nucleus by dividing it many times by mitosis. Then these spores are released. Spores are haploid. They land on a suitable substrate, in this case another piece of bread, and they will grow into a haploid mycelium. 1/2 of these spores are of the plus mating type, 1/2 are of the minus mating type. Okay? Because usually it's just one gene, one allele that determines that. And so this is pretty characteristics of almost all of these zygomycotes. Most of them asexual. But when they do, now something that used to be called a zygomycote that has been separated out, and since I'm not sure, I'll go ahead and put it here, okay? Glomeromycota, okay? These are called arbuscular fungi, and all of these that we know of are in association with vascular plant roots. Okay, so they are non-septate, except for in reproductive structures. So that all three of these are have non-septate mycelium, except for reproductive structures. Okay, and these are endomycorrhizal. Okay, what is mycorrhizae? Okay, this is a general word. If we forget, because we have two different forms, endo and ectomycorrhizae. Okay, the endomycorrhizae. Okay, first of all, let me explain what a mycorrhizal association is. It's a mutualistic relationship between the roots of almost all vascular plants and fungi. The fungi penetrate plant root cells, or they come in association with plant root cells, and they are now an extension of the plant. Okay? Now, this is a mutualistic relationship because the plant gives the fungi photosynthetic material, sugars, amino acids. But what does the plant get out of it? The fungus is better at mobilizing minerals in the soil and water. And furthermore, they make extensive extensions to the roots, and therefore, especially with phosphorus and minerals along that line, the fungi are better at breaking up larger rocks into smaller minerals and cycling of plants. It has been shown that if you grow any of these plants, we're talking about from anything from ferns all the way up to oak trees, and grow them without fungi, their growth is severely stunted. Okay? So there's about 95% of vascular plants have some root association with fungus. Now, endo and ectomycorrhizae are groups of fungi that they don't penetrate the plant cells. They don't have haustoria. Okay, they become associated with the root hair cells, but they don't have projections that go into the cells. Endomycorrhizae do, and these are called arbuscules. It's a type of hypha, haustorial hypha. This penetrates into the root cells, so that is in the cytoplasm of the root cells, whereas the rest of the fungus is outside. Okay, and so that is an apomorphy for this group. Interestingly, we only know asexual stages. No one has been able to find any of these critters that have any sexual reproduction. So they're only producing asexual spores, and these asexual spores are called glomerospores. So all we have here is haploid state for this fungi. But it's obvious that they must have come from ancestors that did have some sort of diploid or dikaryotic state, but we haven't found it in these yet. So this is a fungus in the soil grows between the cell walls, but then it'll penetrate, branch like this, and that's the arbuscule. And so this is endomycorrhizal. Even though it sort of looks like it's parasitic, it's actually mutualistic. Both the plant and the fungus get a benefit from it. Okay, sexual stages are unknown. There may exist, just no one has ever found it. Okay, but based on ribosomal RNA sequences, this is now, we know, the sister group to the rest of the fungi, which is called dikarya. Okay. Now, most of the time, this type of fungus you will rarely see because they never have fruiting bodies that are above ground. Zygomycotes, yes, you see them quite a bit, especially on your bread. Okay, the green stuff, that's the zygomycote. Okay, this is the rest of the fungi, and it's a pretty big group, dikarya, consisting of two major clades. Ascomycota Psidiomycota. And these are far more advanced than other groups. Apomorph for dikarya is that septate. All these down here, non-septate. But all of the higher fungi, we call them higher fungi, they have septa. And the particular types of, instead of producing spores that are called sporangiospores, they have a special type of spore. You can call it a conidiospore or just plain conidia. Now, what is this? Okay, so they're underground. They still send up a sporangiophore, and they'll have hyphae that are sticking out like this, but the spores bud off at the end of these special reproductive hyphae. They bud off from the tips. And instead of being produced inside of a sac-like sporangium, they're being produced off the tips of special hyphae. And so that's a different, and this is mitosis. This is asexual. This is the type of asexual spores that they produce. Okay? All right. In fact, that right there is a good picture. So this is the sporangiophore. These are special reproductive hyphae, but these are the spores that are being produced off of the ends of the hyphae. And those are called conidia, or conidiospores, haploid. And plasmogamy results in a dikaryotic hypha, not just one chamber. So we still have down here, still have the dikaryotic state. This is just general, that we have something that is dikaryotic. But up here with dikarya, it is dikaryotic hypha. In other words, part of the mycelium becomes dikaryotic. All right? It's not just one cell. It's hundreds, thousands of cells that will be dikaryotic. Additionally, okay, so this is extensive. So it's more extensive than just the zygosporangium that you have in Zygomycota. All right? And the fruiting bodies, where sexual reproduction takes place, are macroscopic. Okay, now what do I mean by that? You can usually see it without a microscope. Zygomycotes have fruiting bodies too, but you usually have to have a microscope to really appreciate it, okay, because they're too small. So this is microscopic. This is what you call a mushroom, okay? So these two groups have fruiting bodies that we call mushrooms. And that word mushroom is particularly for the basidial mycota. You can still call these fruiting bodies here also mushrooms, but the real mushrooms are really the basidial mycotas. Okay, so two groups that I already have up here, ascomycota and basidial mycota. Okay. The basidial mycota is very advanced compared to any of the ascomycotas. Okay, now, one problem here in the past of how fungi were divided up was based on their sexual reproduction. Okay, so the zygomycotes produced what they call the zygospore, which is really a zygosporangium, okay? And the ascomycotes, they would produce something called an ascocarp. I'll put that up a little bit later. Basidial mycotes produce something that's called a basidial carp. Those are types of mushrooms. And, but there's some fungi that they put in a class called Deuteromycota, also called imperfect fungi. This is just for historic reasons I'm mentioning this, because they did not have any sexually reproductive structures, and therefore they were just thrown together. Now that we have molecular data, many of these species that don't have sexual phases anymore have been either reassigned to the ascomycotes or the basidial mycotes. So this Deuteromycota is a good example of a polyphyletic group. It's definitely polyphyletic. But penicillium doesn't have sexual reproduction. Okay, this penicillium is that green stuff that's growing on your orange peel. Okay, it has that real fruity smell to it. Okay, but it is a ascomycota based on DNA evidence. Okay, all right. Okay, so ascomycota, they're called cup fungi. Okay, a lot of times, or sac fungi, and that's because their fruiting bodies contains a sac-like cell where karyalchemy will take place, and it's called an ascus. So I'm putting that as the apomorphy for ascomycota. Okay, a lot of times their fruiting bodies will be cup-shaped. There's many different forms, but nonetheless, the asci are the tips of dikaryotic hyphae that are found in that area, in the cup. Some of these may have a fruiting body that has many depressions that look like cups, okay? There's many different forms, but this is where they get their name. And so the whole fruiting body is called an ascocarp, okay? Ascocarp because it contains asci. All right? And this is where the dikaryotic state is found. It's only in the ascocarp for the most part. Okay? The rest of the fungus that's underground is still haploid. Right? So I'll show this here in a moment. So in fact, the ascocarp is sort of complex because we have in the ground, we originally had plus strain, haploid plus strain, haploid minus strain. They grow together and fuse, plasmogamy, producing a dikaryotic hyphae, and all three of these are parts of the ascocarp. So you have some hyphae in the ascocarp that is haploid from the plus strain, some of it's haploid from the minus strain, and the rest of it is dikaryotic. But the layer where the asci are all dikaryotic. All dikaryotic. Okay? I'll show it here in a moment. So, whorls and truffles. Not the chocolate truffles that you buy in the store, that's candy. But this is, French people really like this. It's a very expensive dish. They'll charge you $200 a plate of this because it's hard to find these. Their fruiting bodies do not get up above the ground, and so they train dogs and pigs to smell these things out. Okay, dig them up, but they're considered a delicacy. Okay, so this here is an ascocarp. So I'll go through the life cycle here. And these are other examples of ascocarps. This is a morel. And each one of these little cups that are on here, this is still one fruiting body, but all the asci are inside of those depressions. All right? So what happens in their life cycle? Let's see here. First of all, asexual. Okay. Haploid. And they will produce conidiophore with conidia. Okay? So haploid by mitosis and haploid. However, when you have two mating strains, they can come together and they will produce a particular type of gametangium. Have special names, and I'm not going to give you this, we're not going to worry about the special names, but these will then fuse together. One from the plus strain, one from the minus strain, and what will grow out of that will be a dikaryotic hyphen. Okay? This is not like the zygomycota. Okay? So those gametangia fuse plasmodemy, and then what will grow out of it is a new mycelium. New mycelium that is dikaryotic. That will then form, along with some hyphae from the plus strain and minus strain, a ascocarp. And the lining, I don't like this one, it's a shame. This is the ascocarp, but I prefer that type. They'll have the asci inside. Okay, so here on the cup, yes, let me do this a little bit better. Show what happens. So I have just a little bit more room. Okay, so we have all these hyphae in here, and they come up, and the terminal parts, they will produce, and I'm going to exaggerate the size here, it'll produce these layer of asci. Initially, these asci are dikaryotic. Okay. Then we get karyotic. We have a zygote nucleus. Zygote nucleus in one ascus. This will then undergo meiosis and produce 4 ascospores. These are called ascospores. Each of which will divide once by mitosis. Producing eight ascospores. We still call them ascospores. Eventually, the tip of this ascus will dehiss, that is, it'll break down, and usually under pressure, all these spores are released. So this is happening in just the asci. All these other dikaryotic cells down here, they are sterile. Once again, when I say sterile, they do not participate in reproduction. Okay? So this is what is happening in the ascomycota. And usually they always produce 8 spores. And those spores are called ascospores. Okay, any questions? I will probably, let's stop here, because I can't really finish this story. But the city of Lakota, I'll just give a little introduction here. Most of the stuff you buy in the store that are called mushrooms, and most of the things you see growing outside that you call mushrooms belong to this group. Okay, and it's a pretty big group, and their life cycle will be similar to the Ascomycota. However, the fruiting body is called a basidiocarp, and it produces reproductive cells that are called basidia, which is different than an ascus. So that's Apomorpha here. The reproductive cells are for karyogamy and meiosis are basidia. And it has a long-lived, extensive dikaryotic mycelium that can live for years. Okay, and eventually the haploid state is very brief. Most of the fungus underground, both vegetative and the fruiting body, dikaryotic. Okay, so before I stop here, let me just review dikaryotic state. It occurs down here. All of these up here have something that is dikaryotic. The Zygomycota, it's restricted just to the zygosporangium. In the Ascomycota, it's more extensive. It's in the ascocarp. Okay? Whereas in the Basidiomycota, it's even more extensive, even the vegetative hypha underground, plus the basidiocarp is dikaryotic. And so it's a matter of degree that we're talking about here. Okay? That's where I will stop for now. See what I have on. Yeah, I'll stop here. We'll talk about plant junction. All right. The fungi are interesting because we grow on you.